Hello everyone, I'm Asst.Prof.V.S.Aher, and in the last session of fermentation technology, we have already started with the ethanol production, in which we have already studied the introduction of ethanol, whereas microorganisms and raw materials used for the production of ethanol. So, in our today's session, we'll start with the actual production of ethanol. So, here you can just see a process of production or flow sheet of production of fermentation process. See now, here the basically sugarcane or a raw material, it is being selected, harvested, and then it undergoes the milling process where the sugarcanes are being crushed and a juice is being removed. Further, these collected sugarcane juice, which contains high amount of sugars or carbohydrate source, is transferred through the filtration process. This filtration process, it helps to clarify the sugarcane juice and separate the different types of solid particles which may be present in that juice. Further, this molasses or the sugarcane juice, it is transferred for the fermentation process. Then during fermentation process, here different kinds of raw materials which are being which undergoes pre-treatment, sterilized uh, material have been added. Then the inoculation process is been done. Here we add Saccharomyces cerevisiae strain, which gives the high production of alcohol, and then we start with the fermentation process. Once the fermentation process is completed, we go for the purification, separation and purification process of alcohol. This separation and purification of alcohol, it is being done with the distillation process. With the help of distillation process, pure ethanol has been is been separated, and then further test of this ethanol, it is being carried out, and then it is supplied for the various purposes in for the different uses. This is just a simple flow sheet which explains ethanol production in simple way. Now we'll start with the production process of ethanol in detail. So ethanol production, it can be carried out in three stages. First one is preparation of nutrient solution. Then second step is inoculum development. Third step is fermentation. And then the last step is recovery of the product. In these fourth steps, the fermentation process, it is completed for the production of ethanol. So let's start with the first step, which is preparation of nutrient solution. The most commonly used raw material for the production of ethanol is molasses, which has been extracted uh, or gained from the various sugar industries, or whey grains, potatoes, and wood waste can be also used as a starchy and cellulosic material. When these molasses, it is used for fermentation process, first it is diluted with the help of water. This dilution is done so that the sugar concentration is in the range of 10 to 18%. So the dilution of molasses, it is carried out with the help of water, and 10 to 18% sugar concentration is maintained because this is the higher concentration in which each strain can grow or can tolerate the concentration of sugar. So the dilution of molasses it is carried out. Then if the starchy material it is used, this starchy material, as we have already learned in the last session, it is very important to carry out pre-treatment process of starchy material. So it is first being hydrolyzed by using acidic or enzymatic action. The hydrolysis of starchy material it is carried out, and then they are used as a nutrient in the fermentation process. Then this it can also be done by using barley malt or dilute acids or fungal amylase, production of amylase by aspergillus or rhizopus species is carried out, and with the help of these amylase or diluted acids, the pre-treatment of starchy material it is carried out. Once a nutrients are ready, then we go for the inoculum development. For the development of inoculum, it is very important first to select the microbial strain. Here, as we all know that alcohol production it can be carried out by the yeast or bacterial strain. So once the strain it is being selected, then we go for the isolation of pure strain, and then inoculum is being prepared under aseptic conditions. For this purpose, the organism are first cultured in a flask under aerobic condition. Aerobic condition here it is maintained to increase the size or increase the growth of selected strain. Once the growth is being started, we go on increasing the inoculum, which can be used for the inoculation process in the large scale fermenters. So once the nutrients are ready and inoculum development is completed, then we go for the third step, which is the fermentation process. Here basically batch fermentation process is being used on large scale for the production of alcohol, but nowadays continuous fermentation process are also being utilized for the production of alcohol. It has been possible to increase the alcohol production by 10 to 12 folds by continuous fermentation as compared to batch fermentation process. Previously when we were using batch fermentation process, as compared to batch fermentation process, 10 to 12 times larger yield can be obtained in continuous fermentation process. So nowadays continuous fermentation process is used on the large scale for the alcohol fermentation. So industrial production of alcohol or ethanol it has been carried out in the large fermenters up to size of 1,25,000 gallons. So in these large scale fermenters, nutrients are added, and pH of this medium it is maintained around 4 to 4.5, and the initial temperature it is kept between 21 to 26 degrees Celsius. As ethanol gets evaporated at 27 degrees Celsius, so it is very important to maintain the temperature below 26 degrees Celsius. Initially the aeration is done in the fermenters for the proper and good growth of the organisms. Later, anaerobic conditions are created by the withdrawal of oxygen coupled with the production of carbon dioxide. So initially when we start the fermentation process, pH is adjusted between 4 to 4.5 and temperature is between 21 to 26 degrees Celsius. So once the fermentation process is been started, we supply the oxygen by supplying sterile air inside the fermenter. And this helps in a proper growth of the organism. Once the growth of organism have been started, the oxygen it is been removed from the fermenter, and side by side, as the fermentation process is been started, the sugars, the organism start converting the sugars into alcohol and carbon dioxide, and the production of carbon dioxide is responsible for the development of anaerobic conditions inside the fermenter. It takes 2 to 3 days for the fermentations to be completed in of the production. As fermentation is completed, the fermentation broth contains ethanol in the range of 6 to 9% by volume. Then it states that, or it represents that, about 90 to 95% of substrate is being converted into ethanol. Once our fermentation process has been completed, around 6 to 9% by volume ethanol production have been detected, which indicates that 90 to 95% of substrate is been completely utilized for the production of ethanol. Here you can see the simple reaction in which glucose is converted into ethanol and carbon dioxide. The one molecule of glucose can produce two molecules of ethanol and two molecules of carbon dioxide. So once the fermentation process is completed, we go for the recovery of ethanol, which is separation and purification of ethanol. The cell mass or the organisms present in the broth, they are separated by the centrifugation or sedimentation process. Ethanol from the fermentation broth, it can be recovered successively by the distillation process. For this, concentration about 95%, if it is present inside the fermentation broth, special technique of distillation is been adopted. Here we prepare absolute 100% alcohol for the distillation process, and azeotropic mixture means a mixture of water, benzene and alcohol, it is prepared. Then this mixture of benzene, water and alcohol, which is known as azeotropic mixture, it is distilled by gradually increasing the temperature. So by this technique, it is possible to first separate benzene, ethanol and water mixture. It first removes benzene, which has low evaporating point. It first removes benzene, then it removes ethanol, and then it removes water. Then ethanol and benzene mixture, it is removed, and the remaining is the absolute alcohol. And in this way, the recovery or the separation of alcohol can be carried out 100%. Here you can see one more flow sheet which shows the ethanol production using sugarcane. The sugarcane, they are once harvested, milling process is done, then the sugarcane juice, it is extracted, and this juice is then treated with the Saccharomyces cerevisiae in the fermentation process, where it produces alcohol as well as carbon dioxide. Then it undergoes distillation process. After distillation process, we may carry out molecular seize, the filtration process, and these anhydrous ethanol as well as distilled alcohol, it is used for the wineries and other industries. and the remaining waste material, which is known as bagases, they are used in the various boilers for the heat, steam, and electricity generations. Various alcohol industries have their own electricity production plant also, in which they use the waste raw material or the bagases which are separated as a waste material in the industries. Here, it is another flow sheet in which corns or the starchy materials which are used for the production of alcohol. The corns or other malts are being soaked. Their malting and mashing process is done. Grinding, cooking, liquefying, and then saccharifying. This addition of sugar is done, in which various types of enzymes and yeast strains act on this starchy material, and with the help of this yeast strain the fermentation process is completed. Once the fermentation process is completed, then we go for the recovery by the distillation centrifugation process, in which grains are being separated, various distillers solubles are being separated, and in the distillation process we separate ethanol and the CO2. So this is all with about the production of ethanol. Now once the ethanol is produced, it have the high demand in the market. We'll see the applications of ethanol. It is used as a solvent or chemical intermediate or fuels. Now solvents in various resins, pharmaceutical industries, cosmetics, household cleaning products, industrial solvents, ethanol is used in a large scale. Then as a fuel in or as a biofuel in the internal combustion of engines, ethanol is being used on the large scale, whereas in various petroleum derived chemicals or butadiene production, as a chemical intermediate, ethanol is being used. So this was all about the ethanol production and recovery. Thank you for watching.